Axolotls, the salamanders that can regrow limbs, spines, and even brains. They are small, aquatic, and perpetually youthful. With wide smiles and feathery external gills, axolotls may seem like charming curiosities from a distant lake in Mexico, but beneath their delicate skin lies a biological capability that has fascinated scientists for decades. Axolotls possess one of the most astonishing powers in the animal kingdom, the ability to regenerate entire limbs, spinal cords, portions of the brain, heart tissue, and more, not just once, but repeatedly, throughout their lives. Among vertebrates, no other animal regenerates with such speed, precision, and completeness. When an axolotl loses a limb, something remarkable happens. Instead of forming a scar as humans would, the wound remains open for a short period before being sealed over by skin cells that don't trigger fibrotic healing. These cells begin to revert to a more primitive, undifferentiated state. This process, known as dedifferentiation, leads to the formation of a regeneration-specific structure called the blastema. The blastema functions like a developmental time capsule. It is packed with progenitor cells that carry the potential to become bone, cartilage, skin, blood vessels, muscle, and nerves. The formation of the blastema marks the beginning of a perfectly choreographed sequence of cellular events that results in a fully regrown and functional limb. Crucially, this regeneration doesn't occur in isolation. The process is deeply dependent on communication between different tissue types. In the early stages of limb regeneration, nerves play an essential role. Without nerve input, the regenerative process stalls. Neural signals interact with epithelial cells covering the wound site, triggering a cascade of growth signals that sustain and guide the formation of new tissue. These interactions aren't chaotic, they're governed by something that makes axolotl regeneration even more incredible, the ability to know exactly what's missing and what needs to be rebuilt. This remarkable precision stems from a built-in system of positional information. Cells within the blastema carry molecular cues that tell them their location along the limb axis, whether they should become a fingertip, a wrist, or a shoulder. Even when parts of the blastema are rotated or transplanted, the regenerative system reorients and repairs the pattern correctly. This spatial awareness is regulated in large part by a gradient of signaling molecules, the most studied of which is retinoic acid. Retinoic acid, a derivative of vitamin A, plays a central role in orchestrating axolotl limb patterning. Its concentration is highest near the body and decreases toward the limb's distal end. This gradient provides clear positional instructions to blastema cells. Experimentally manipulating retinoic acid levels can even confuse the system, inducing the regeneration of extra limbs or causing limbs to regenerate in the wrong configuration. It is the molecular equivalent of giving cells a map, and it works with astonishing accuracy. But positional control is only one aspect. For regeneration to proceed rapidly and efficiently, axolotls rely on an optimized cellular machinery that enables high-speed protein production. A key player here is the mTOR pathway, a universal biological switch that governs cell growth and protein synthesis. Axolotls possess a hypersensitive version of the mTOR protein that allows them to switch on protein production almost immediately after injury. Their cells also maintain a reservoir of pre-transcribed messenger RNAs, like a stockpile of blueprints ready to be activated. This means that once the blastema forms, the cells are equipped to start building, without waiting for the genetic instructions to be copied from DNA. At the same time, axolotls avoid the greatest enemy of regeneration, scar tissue. Unlike mammals, axolotls exhibit neoteny. They retain juvenile characteristics throughout life, including an extracellular matrix and immune system configuration that supports regeneration instead of scarring. Their immune response is carefully balanced, enough to protect from infection, but not so aggressive that it leads to fibrosis. In humans, the inflammatory response to injury tends to result in dense, fibrous scar tissue, which blocks cell migration and tissue patterning. Axolotls skip this step entirely. The result is perfect tissue regeneration with no permanent marks. So why can't humans do this? The frustrating truth is that humans share many of the same genes and molecules involved in regeneration. Retinoic acid plays a major role in our embryonic development. The mTOR pathway is fundamental to our cellular metabolism. But after birth, and especially after fetal development, our cells lose the ability to re-enter these regenerative programs. Instead, our healing is geared towards speed and protection, sealing wounds quickly, often at the cost of functional recovery. Scar tissue forms fast and permanently, a natural but limiting solution. That's why scientists are turning to the axolotl. By studying its molecular pathways, researchers hope to unlock ways to reduce human scarring, stimulate tissue regeneration, and perhaps even one day, regrow human limbs. Several avenues are already being explored. Some researchers are experimenting with drugs that modulate the mTOR pathway, aiming to enhance regenerative activity in mammalian cells. Others are working on gene therapy approaches to replicate the effects of axolotl proteins in human tissues. The medical implications go far beyond limb loss. If we could harness even a fraction of the axolotl's regenerative abilities, we might treat spinal cord injuries, repair damaged heart tissue, restore sections of the brain lost to trauma or stroke, and dramatically improve organ regeneration. Even skin healing, one of the simplest forms of regeneration, could be revolutionized. In fact, some commercial products have already been inspired by axolotl biology, such as collagen-based skin treatments that mimic the extracellular environment of axolotl skin to promote scarless healing. Scientific progress in this field is accelerating. New genetic tools, such as CRISPR gene editing and single-cell sequencing, allow scientists to map the molecular choreography of axolotl regeneration in unprecedented detail. Whole genome studies have identified key regulatory elements that control regenerative responses. Cross-species comparisons are helping researchers understand which components are conserved in humans, and which have been silenced or lost over evolutionary time. The axolotl is not just a scientific curiosity. It is a roadmap. A living blueprint of what is biologically possible. It reminds us that nature has already solved many of the challenges we face in medicine. We only need to observe, understand, and adapt. As researchers continue to decode the axolotl's secrets, we come closer to a world where lost limbs, spinal cord injuries, and damaged organs are not irreversible conditions, but temporary setbacks with biological solutions. What once seemed like science fiction is slowly becoming science fact. And it all begins in a quiet lake in Mexico, with a smiling salamander that refuses to grow up, and refuses to accept the word permanent. Tardigrades, the microscopic creatures that survive in space. Tardigrades are among the most extraordinary life forms ever discovered, microscopic, eight-legged animals that defy the very limits of biology. Barely half a millimeter in length, they may look unassuming, even alien, with their pudgy bodies and clawed limbs. Yet they possess a level of resilience that no other known organism on Earth can match. These tiny creatures can survive the vacuum of space, doses of radiation that would obliterate humans, and temperatures near absolute zero. But how is this possible? How can such a simple organism endure environments so hostile that they seem incompatible with life? To understand the tardigrade's incredible toughness, we must first look at its defining survival strategy, cryptobiosis. When faced with environmental extremes, such as intense heat, freezing cold, dehydration, oxygen deprivation, or exposure to radiation, tardigrades perform a remarkable transformation. They enter a dormant state called cryptobiosis, in which they lose almost all of their body water, shut down their metabolism to just 0.01% of normal activity, and curl into a compact, desiccated form known as a ton. In this state, they appear lifeless. But in truth, they are in suspended animation, waiting for favorable conditions to return. This ton state is the key to their survival across extremes that would destroy most other life. Inside the ton, tardigrades employ a suite of molecular tools that stabilize and protect their cellular components. One of the most remarkable adaptations is the production of a group of molecules known as intrinsically disordered proteins, TDPs. These proteins do not assume a fixed three-dimensional shape like typical enzymes or structural proteins. Instead, they remain flexible and unstructured until dehydration triggers them to form a glass-like matrix around the cells. This matrix preserves membranes, organelles, and even proteins themselves, essentially freezing the internal structures of the organism in place, preventing collapse or rupture. TDPs act like molecular scaffolding during cryptobiosis, creating a safe harbor for life within a body that has shed all liquid. But dehydration and physical stress are only part of the challenge. In the vacuum of space or under conditions of high radiation, DNA damage becomes inevitable. This is where another tardigrade innovation comes into play, a protein known as TSUP, short for damage suppressor. TSUP is unique to tardigrades and was only recently identified through genome sequencing. It binds directly to DNA and shields it from the double-strand breaks caused by reactive oxygen species and ionizing radiation. In combination with highly efficient DNA repair mechanisms, TSUP allows tardigrades not just to avoid damage, but to recover from it. This two-pronged defense, protection and repair, makes their genetic material nearly indestructible, even under the most punishing conditions imaginable. These adaptations are not theoretical, they have been tested in the most extreme lab known to humanity, outer space. In 2007, tardigrades were launched into low Earth orbit aboard the European Space Agency's Photon M3 mission. They were exposed directly to the vacuum of space, unshielded from cosmic radiation and ultraviolet rays. After 10 days, many of them were still alive. Some had even reproduced. This was the first confirmed case of a multicellular organism surviving the harsh conditions of space, proving that tardigrades are not just resistant, they are space-worthy. And their talents don't stop there. Tardigrades have survived temperatures as low as -272 degrees Celsius, just 1 degree above absolute zero, and as high as 140 degrees Celsius. They have endured pressures greater than those found in the deepest ocean trenches and have walked away from environments devoid of oxygen. In all these cases, it is their ability to enter and exit the ton state that makes the difference. When conditions return to normal, the tardigrade rehydrates, reactivates its metabolism, and resumes life as if nothing had happened. This cycle can repeat many times, and individual tardigrades have been known to revive after years in dormancy. It's important to note that tardigrades did not evolve these abilities to survive space. These adaptations are thought to have evolved on Earth in response to extreme terrestrial environments, such as dry mosses, alpine rocks, desert soils, and temporary freshwater pools that regularly dry out. In these habitats, organisms must withstand cycles of dehydration and rehydration, freezing and thawing, and high levels of UV radiation from unfiltered sunlight. The traits that allow tardigrades to persist in these environments also happen, incidentally, to make them the toughest organisms for space travel. In other words, they are a triumph of Earth's own evolutionary pressures, accidental astronauts shaped by moss and soil. What tardigrades teach us goes beyond biological curiosity. They offer clues about the very limits of life, where it can exist, how it can adapt, and how it might one day spread beyond our planet. Some scientists are exploring how tardigrade genes could inform new medical technologies, such as stabilizing vaccines without refrigeration, or protecting human cells during radiation therapy. Others are studying TSUP to see if it can be used to protect DNA in synthetic biology or spaceflight applications. The potential is vast, and we are only beginning to tap into what these tiny animals have to offer. Their biology also forces us to rethink what life needs. If a creature can live without water, without oxygen, and without pressure, if it can pause its life for decades and resume it as if nothing had happened, then what other forms of life might be out there in the cosmos? What other organisms, on other worlds, might exist not in the warm, wet conditions we consider typical, but in places far more extreme? Tardigrades don't answer these questions directly, but they open the door. They remind us that life, when pushed to the edge, doesn't necessarily break. Sometimes, it adapts. Sometimes, it changes form. And sometimes, it survives in ways we once believed impossible. So the next time you look up at the stars and wonder what kind of life could be out there, remember that here on Earth, one of the toughest, most space-ready creatures is already with us, hiding in droplets of moss, crawling beneath leaves, waiting for the next rain, or the next decade, to begin its life again. Tardigrades may be microscopic, but they carry within them one of the boldest statements life has ever made: I can survive anywhere. Mudskippers, the fish that walks, blinks, and breathes on land. They live in the in-between, where land meets sea, and mud becomes the stage for one of evolution's most fascinating performances. Meet the mudskipper, a fish that walks, breathes air, climbs trees, and thrives in an environment few vertebrates would dare to call home. Mudskippers are members of the subfamily Oxydercinae, a group of amphibious gobies that have redefined what it means to be a fish. Found across the intertidal mudflats and mangrove forests of the Indo-Pacific and the Atlantic coast of Africa, these creatures are anything but ordinary. While most fish remain prisoners of water, mudskippers have broken free, living large parts of their lives on land. But how does a fish conquer the world above water? It starts with their movement. Mudskippers have evolved powerful, jointed pectoral fins located near the front of their body. These fins work like crude arms, pushing them up and forward as they crutch across muddy terrain. The motion resembles a hybrid between crawling and skipping, an awkward yet remarkably effective mode of terrestrial locomotion. Some species can even use this technique to climb onto low-hanging branches and exposed roots, expanding their territory vertically in a world ruled by gravity. Their eyes are another marvel. Protruding from the top of their heads like tiny periscopes, these large, bulbous eyes give mudskippers panoramic vision. Each eye can move independently, scanning for both predators and prey on land and in the water. But here's the twist: mudskippers have developed a unique way to keep their eyes moist and protected while out of water. They retract them into water-filled sacs, effectively blinking. This blinking mechanism is incredibly rare among fish and shows striking parallels to terrestrial animals. Breathing is yet another problem that mudskippers have solved with elegance. Although they do not possess lungs, these fish can absorb oxygen through the moist surfaces of their mouth and throat, a form of cutaneous respiration. They also store water in their gill chambers, keeping their gills functional even during extended trips on land. For some species, like Periophthalmodon schlosseri, access to air is not optional, it is essential. Without it, they drown. And if that's not enough, consider their skin. Thick, mucus-covered, and water-retentive, their skin helps prevent desiccation. It's also highly resistant to disease, providing a protective barrier in a world teeming with microbes, parasites, and pathogens. This armor allows mudskippers to remain active for hours at a time on land, hunting insects, defending territory, and even courting mates, all without ever needing to dip back into the water. Mudskippers are also known for their bold, sometimes comical behaviors. They're territorial, often squaring off in open-mouthed displays and tail-slapping duels on the muddy battlefield. Their burrows, dug deep into the soft sediment, serve as shelters from predators, places to trap air during high tide, and nurseries for their young. These burrows can reach impressive depths and are often oxygenated, creating a life support system that bridges the underwater and terrestrial worlds. But these fish aren't just biological oddities, they are ecological engineers. By constantly digging, aerating, and moving sediment, mudskippers play a vital role in maintaining the health of intertidal ecosystems. Their presence impacts nutrient cycling and influences the distribution of other organisms. Because they are highly sensitive to pollution, erosion, and habitat loss, mudskippers are considered excellent indicators of environmental change. When the mudskippers vanish, it's often a warning sign that something is wrong with the ecosystem. And then there's the evolutionary question: what can mudskippers teach us about the leap from sea to land? The answer is, a lot. Their bodies provide clues to the kinds of adaptations early tetrapods may have needed to make the transition from aquatic to terrestrial life. Features like eye blinking, air breathing, limb-like fins, and behavioral versatility are all part of this story. Scientists studying mudskippers have discovered gene expression patterns that mirror those found in amphibians, as well as specialized immune responses, hormonal shifts, and anatomical changes that illustrate the extraordinary plasticity of vertebrate evolution. As Dr. Fabienne Ziadi-Kunzli of the Okinawa Institute of Science and Technology puts it, they are fish, but they can walk and live partly on land. Mudskippers are not just survivors, they are innovators. In a world that constantly changes, they have become a symbol of adaptability. They challenge our assumptions of what a fish can be, and they remind us that the boundary between water and land is not a wall, it's a door. And through that door, the mudskipper skips, crawls, and climbs, writing one of nature's most unforgettable stories.